we said, I am a diabetes specialist, run a diabetes unit in KEM hospital for last 35 years. And I have concentrated on mother and baby as a unit which predisposes to future diabetes. This is the only diabetes unit in the world where the logo is mother and baby. We have acquired a new logo, that's the modern Ganesh. And when we were children, he used to travel on the back of this mouse. Now he clicks the mice and moves very fast. My unit combines the research on intrauterine origins and rapid transitions as basic causes of diabetes in India. We have many collaborators, including CCMB, which does all our molecular biology, and some institutes abroad. I wrote my confessions a few months ago. I have been called internationally as a thin, fat Indian because that's how I described as Indians. And that became popular when I published my picture in Lancet about 15 years ago. Myself and my English friend, we both had identical BMI of 22.3. I have been blamed throughout my life for being thin. John's body fat percent on a DEXA machine was 9%. And 15 years ago, my body fat percent was 21%. For same BMI, we had very different adiposity. And that's how I described the thin fat Indian. Today, a third of what you see is fat. So if you find some way of dissolving me, you will see almost a third of fat dissolving here. The dogma which we learned for diabetes as students is that there is a genetic susceptibility and it's precipitated mainly by obesogenic factors. Today we know it's polygenic and therefore, at least in today's world, it's non-modifiable. So prevention of diabetes, which should be the goal, actually has taken help of manipulating diet, physical activity, or correcting the inactivity and modifying stress as part of prevention strategy, whether it be in the American diabetes program or in the Finnish diabetes or the Indian diabetes program. However, an average participant in these trials is 55-year-old. Usually they are obese, 34 BMI, and they have impaired glucose tolerance, which is pre-diabetes. 3 end-stage factors: age, obesity, and glucose intolerance. And still they have been calling it primary prevention trial. My submission is that actually it's treatment of precipitating factors and nothing to do with the susceptibility. It also happens post-reproductively, and therefore the benefit is only for the individual who has been treated. The advice has been eat less and walk more. And I'm going to show you that there might be a modifiable susceptibility. And at least in India, if young women were to eat less and walk more, then we will exaggerate the diabetes epidemic. Story started many decades ago in Netherlands when Germans cut off the food supply to Western Holland towards the end of the Second World War. The babies born to pregnant women that time have been followed. These babies faced what we called intrauterine famine. If they faced intrauterine famine in the 3rd trimester, they were less obese than children who had not faced the famine. While if they faced the famine in the first 2 trimesters, they were more obese as young adults, indicating that intrauterine exposure to famine had an effect on their risk of obesity, and the windows in pregnancy actually gave you different results, either increased or decreased susceptibility. Around same time, reports from Pima Indians, which is one of the very obese populations in the world, showed that if mother was diabetic during pregnancy, her children were more likely to be obese either in early childhood, during adolescence, or adults, compared to the children who were born before the mother became diabetic. It's the same mother, same genetics, but if mother had diabetes in pregnancy, intrauterine hypoglycemia, then baby was at a higher risk of diabetes. These two studies highlighted that intrauterine nutrition of the baby acted over and above the genetic susceptibility to either decrease or increase the risk of diabetes. 1991, this phenomenal paper was published in BMJ by David Barker and Nick Hales, and they showed that in a cohort in the Britain, birth weight was inversely related to risk of diabetes as adults. So that people who were born with lower birth weight had higher risk of diabetes. David Barker came and met me in February 1991 just to convince me of this, and that changed my life. In the first meeting, I suggested to him that he calls his hypothesis 50 phenotype, and he actually did that. And that hypothesis says type 2 diabetes is the outcome of the fetus and early infant having to be nutritionally thrifty. And he suggested that this programmed the fetus in a way that he became, or he or she became at increased risk of diabetes in future. A very interesting support came when my Danish colleagues studied identical twins in Europe and showed that smaller of the monozygotic twins had a higher risk of diabetes. Same genetics, but intrauterine exposure to undernutrition, small size at birth, increases the risk of diabetes. And then came the concept which you all are very familiar with, but as clinician I wasn't, of plasticity and programming. Of course, a developing system is plastic. It has the ability to develop in different ways, provided there is an adequate and good environment. And it also says that during development, if you are exposed to a particular environment, it remains with you lifelong because that's how the blueprint is laid down. It affects the structure and function. If there is an adverse or a related or adversity related restriction of this plasticity, that's what we call programming. And that's now a buzzword in clinical medicine. Windows of vulnerability for such sort of restriction of plasticity. The most prominent one is pre and periconceptional period, pregnancy itself, lactation, adolescence, when there is a renewed growth and development in the body. It could have genetic and certainly some epigenetic mechanisms as the cause of it. And the environment which has been investigated, especially in animal models and tested in humans, includes nutrition, maternal metabolism, stress, a number of pollutants, but the list is increasing. The effects usually last multi-generationally. And there is a possibility of reversibility. And yesterday evening, we were told that meditation might actually deprogram you. So I think the society thought of putting all these together, creating a background for my talk. So we developed this concept of DOHaD, Developmental Origins of Health and Disease. And it says that what we see in adults as disease actually starts developing very likely intergenerationally but not genetic. And it has effects and windows across the life course where a number of changes predominantly in the epigenome affect the growth, development and differentiation, leads to varying phenotypes and finally ends up in the disease. So pregnancy then becomes experience for the two, for the mother as well as the baby. There's one situation where it becomes experience for three. And you again perhaps know more about it than I do. If a woman is pregnant with a female child, the ova are all present at birth. And these ova are exposed to maternal environment from periconceptional or maybe a little post-conceptional period throughout the pregnancy. So there are 3 generations within this pregnancy: the mother, her daughter, and her grandchildren. And again yesterday we saw Srimati Rajam and her Hirza, and I don't need to convince you more on this. So fetal programming actually means so many things which we have assumed to be parts of different, like say, societies or different specialties. So there is, of course, the organism. As a clinician, I study him. I study his systems and organs, study his tissues, and this is where all molecular biologists, biochemists, and other colleagues of mine contribute. So it's a spectrum along the whole from molecule to the human being. Then, of course, there are well-known mechanisms which are known, and that would, of course, include a number of epigenetic mechanisms like DNA methylation, histone modifications, and the new, like say, the part of genome which does not transcribe into miRNAs and other actions, which are now being sort of repeatedly shown to be associated with these epigenetic phenomena of the programming. So the dogma now we could rewrite as that there is a susceptibility which may be genetic, but in addition there is a modifiable epigenetic susceptibility. And this changes the whole scene, because we can now think of preventing diabetes through the modifiable susceptibility if we act very early and probably intergenerationally. And therefore, peri-reproductive period becomes a very important issue in prevention of non-communicable diseases. Now, this is an interesting cartogram. Cartogram is a world map where size of the country is shown proportional to the burden of the disease. And it's a very interesting thing. This website, worldmapper.com, has hundreds of them. So as a diabetes specialist, I used to show this one. And we call India as one of the capitals of world diabetes. And that's how fat India and China are. But interestingly, India is even fatter for undernutrition in early life, both if you measure low birth weight or under 5 undernutrition. And this is contributed both by micronutrients and macronutrients, which are deficient in young girls, and that's how our babies look at. We also written a nice paper on evolutionary perspectives of increased risk in India with Jonathan Wales, who is an evolutionary biologist in London, Barry Popkin, who is a world famous obesity expert, and anthropologist from Pune, Dr. Warimbe. I've just chosen one figure from that paper to show you something which is quite dramatic. This graph shows the height of the world populations over 150 years between 1830 to 1980. Europeans gained about 15 centimeters of height in these 150 years. Indians are running flat. And some of the Southeast Asians have actually even lost height. This is very interesting because this tells us what baggage we are carrying with us. We have been undernourished, stressed from environment for generations, and after 1980, what's happening, I'll show you because that's around the time I started my research. This paper we have published recently as part of Global Burden of Disease in India in Lancet. There are 5 papers. I think you should all read it. They are about non-communicable disease and communicable disease epidemiology in India between 1990 and 2016, so over a 25-year period. This was the prevalence of diabetes in different states in 1990. This is what happened in 2016. Color, when it becomes more red, indicates higher prevalence. And this, interestingly, is the change in prevalence. And increase of diabetes is mostly in the states which we call Bimaru states. It's the relatively undernourished and heavily populated states of India which have shown the highest incident increase in risk of non-communicable diseases. This is for diabetes. So again, we come down to genes, environment, and on this background, when I wrote this paper in 2008, it was very interesting to think about it. This is my friend Andrew Hattersley, who discovered the FTO gene for obesity. He described that FTO gene, which was actually found out on the jivas of type 2 diabetes, increased risk of diabetes by about 25%. If you adjusted it to BMI, the risk actually became non-significant, and therefore it was sold as obesity gene. We were part of this study. We showed that Gerraj and myself that the risk for diabetes was same, 25% increase, but you adjust for BMI and no change in the risk, indicating that genotype-phenotype association was different in India. So overall, I actually now have come to believe Soren Kierkegaard that life can only be understood backwards. And I think this is a profound thing which he said almost many hundred years ago. And if you look at the research in my unit over the last 30 years, though we are now actually studying diabetes, I started in adults, then went to children, then went to pregnancy, and then now we are intervening before conception. So that's how I have started looking backwards. The very first study I did with David Barker and colleagues, and since that he has been our collaborator, we studied over 400 children born in my hospital on whom we had birth weight records from the labor room. And we studied their oral glucose tolerance test at 4 years of age. And we showed what David Barker had predicted. There was an inverse association between birth weight and glucose and insulin concentrations during the test. Once I was convinced what David was saying was true, I said, I'm going to now join you full time. We followed these children at 8 years of age and made an additional discovery. At 8 years of age, we could see that children who were born the smallest, that's the first tertile of birth weight, this line, but who had grown the heaviest at 8 years, this is the highest tertile, they were at highest risk of diabetes. They had higher adiposity, central adiposity, higher glucose, higher blood pressure, higher lipids, and were the most insulin resistant. Being born small but becoming big was the highest risk for non-communicable disease. And what do we do when a child is born small? The parents, the doctors, and the community tries to make them normal. If you try to actually make them bigger than what they should have been, that actually increases the risk of diabetes and non-communicable diseases. Now we have a 21-year follow-up on them. And what was here shown as a risk factor has now converted into real risk of diabetes. By 21 years, we are able to show that those who are born the smallest but have grown the biggest are now either pre-diabetic or diabetic. So we have traced the whole life course story of birth weight and growing subsequently. Then we decided we must know more about why Indian babies are small. As I told you, Indian babies are the smallest in the world with an average birth weight of 2.7 kg. So we set up this preconceptional community-based cohort in 6 villages near Pune in an area called Vadu, where the dynamic sort of chief medical officer of my hospital, Mrs. Banu Koyaji, had set up a huge community-based program. We went to 6 villages, studied over 2,500 women. They were married but not pregnant. And we followed them through pregnancy, then the childbirth, and for now 20 plus years. We have measured them extensively in pregnancy, at birth, every 6 months after birth, every 6 years after birth. We do their metabolic and cardiovascular risk assessment. And we have an extensive biobank starting from beginning. And I know you are going to ask me, we have now added microbiome to that. So there are enormous possibilities. The average mother was 42 kg to begin pregnancy with a BMI of 18.1. And the average birth weight of the child was 2.7 kg. 70% was small by international standards. Interestingly, this 2.7 kg baby born in Pune villages, when it was compared with 3.5 kg baby born in Southampton, had higher adiposity. It looked thin, but it was fat. People never believed our anthropometric story. 5 journals rejected it. So you have to play their game. I did whole body MRI of newborns. And we could demonstrate that when we compared 30 babies in Pune with 30 babies born in Imperial College in London, Indian babies had higher fat in all 3 compartments of abdomen. This actually established the concept of thin fat Indian baby. That means whatever we are seeing in adults has happened intrauterine. It's present at birth. If you want to prevent it, you need to go backwards. We also showed association of red cell folate concentrations with fetal growth, and higher homocysteine predicted intrauterine growth restriction. That was driven largely by vitamin B12 deficiency, which was very common in this population, folate deficiency almost non-existent. And then we have followed up these children. Now we are going to start next week their 24-year follow-up. And we showed that intrauterine exposure to vitamin B12 and folate actually increase the risk of their diabetes in future, so that those who had faced the lowest B12 concentration in the mother in pregnancy and highest folate concentration were at higher risk of being insulin resistant or adipose. In other words, an imbalance between these 2 very related vitamins, both control 1-carbon metabolism, actually had done something which increased their risk. Our national program provides folic acid to adolescents and pregnant women because we have borrowed that from Western world, which are non-vegetarian and B12 adequate. There is no consideration of B12 in our policy. And low B12 and high folate seems to be a combination which predicts diabetes risk in future, either through insulin resistance, adiposity, and we have number of studies showing their neurocognitive effects. At 18 years, half of these are still underweight, BMI less than 18.5%, 5% are overweight, 10% are stunted, but 28% of them have pre-diabetes. 35%, 37% of boys and 18% of girls. This is a very, quote, undernourished population in villages with one of the highest pre-diabetes prevalence ever recorded. And therefore, for me, I've got more and more interested in one carbon metabolism, which is involved in all different aspects of nucleic acid biology. Won't go into details of that, but that's what has actually excited us. And with Giriraj's help and his PhD students, we have published quite a bit. And we have shown that B12 supplementation somehow or other affects methylation of diabetes-related genes, that jivas of B12 deficiency showed hits which are involved in host microbial interaction. And with Yogesh Shauche, we have shown microbiome of either diabetes and showed that Indian microbiome may be different in Western microbiome in many ways. This is the mother in 1993 to 96. That's her daughter. She is here 20 years later. This is the rapid transition. And what we are able to do now, for the first time in the world, that we had F0 mother's pregnancy metabolism recorded. Child was born. That child has been metabolically typed 3 times up to early adult age. 125 of these girls are now married, pregnant and delivered. And we have a glucose story across 2 generations. No other unit in the world has this. And we have now been able to model this to show that what we see in this girl's pregnancy here was already programmed in her mother's pregnancy 25 years ago. These curves are all tracking. So I think this is one of the most rewarding pieces to show intergenerational tracking. I formed this hypothesis about 2010 or a little earlier than that 2 cycles are running. One of fetal undernutrition and other of fetal overnutrition, predominantly by maternal diabetes or obesity. And in India, the 2 are really merging now because of the rapid development, and we are still carrying the stigmata of nutritional deficiencies, but suffering from increasing obesity and gestational diabetes, and we are contributing to diabetes both from the low as well as the large birth weight. So I'll say that conventional models of diabetes had genetics and adult lifestyle as the components. DOHaD now tells us that there is a life course evolution and there are windows of opportunity which are modifiable. Preconception and intrauterine period seems to be very important. There are modifiable epigenetic susceptibilities. Rapid transition and double burden of malnutrition has contributed to the increased risk, and intergenerational solutions for prevention are perhaps to be entertained now. When the progress is the problem, it's difficult to convince everyone. And it's very difficult to tell people that actually the progress is contributing to your morbidity and mortality. And this is where social scientists, anthropologists, and others can help us. Whole lot of people, collaborations across the world, and I think I have to thank all of them, especially the participants who are a very interested bunch who have helped us for last 20 years. We have follow-up rates of more than 95%, which is the best in the world for any community-based study. And we have been funded by Wellcome Trust continuously for 25 years, MRC of the UK, DFID, DBT India, ICMR, DST, NIH USA, and the Nestle Foundation. Thank you.